Cracking the genetic code took a lot of work, as you can see in the classic papers of Nirenberg, Matthaei, Leader, and others. But this is some of my favorite, most elegant biochemistry ever, and I want to tell you about it.
So what do I mean by like cracking the genetic code? Basically, I mean figuring out which mRNA letters, so which messenger RNA letters spell which amino acid or protein letters. So basically, how do you go between the language of nucleic acids, so like the genes and the messenger RNA copies of them, and the proteins? So how do cells know what protein to make from a genetic sequence? And so we now know that it's determined by these like 3-letter words or codons that I'll talk about. And basically what happens is that in the messenger RNA, a codon will show up and a transfer RNA is going to come and it has the matching anticodon and it has the corresponding amino acid and it's going to add that and you keep going and keep going. We now know that these codons are 3-letter words. And so each 3-letter word is going to spell a single amino acid. And so a transfer RNA is going to bring the corresponding amino acid. And now we know that which all of these amino acids, which codons spell for what amino acids. And this is determined through these experiments I want to tell you about. The instructions for making proteins are written in the form of genes, which are DNA. Regions of DNA. And then in a process called transcription, this DNA, an RNA copy of this gets made, we call this messenger RNA. And these copies are used by this ribosome, this protein making complex, travels along and makes a protein. And we know now that how it makes a protein is basically it's reading the messenger RNA in like 3-letter words or codons. So 3 letters is going to spell one amino acid. And the corresponding amino acids, these are the protein letters, it's going to be brought by a transfer RNA, a tRNA, which has a complementary anticodon. So this is going to basically like pair with this codon in the mRNA. And since it's attached to the corresponding amino acid, now you have the right amino acids added. And the protein can then the ribosomes can shift over. The next transfer RNA can come that matches this codon and has this a matching anticodon, and so on and so forth. So we know all of this now, but they didn't know that at the time. And so we're talking about experiments that are going on in the 1960s when all of this is really being figured out. We now know that there are these 3-letter words. So that we have 20 like genetically encoded common amino acids, and we have 4 DNA letters and 4 RNA letters. And so they didn't know if it was like 2-letter words, like were codons 2 letters, were codons 3 letters, were codon 4 letters. They didn't know this, and so they knew that there was this connection between the like messenger RNA. Well, actually, they actually had to figure out that messenger RNA really was a thing, and I'll show you more on how they did that later. But once they did, they didn't know like how the messenger RNA was corresponding to a protein. Like, how did the ribosome know what to add? And so actually this is like they're just starting to learn about like these transfer RNAs and that sort of thing. But even then, they don't know, like, is the code in 2 letters? Are the words 2 letters, 4 letters, 3 letters? And so they had some hints. So they had, Marshall Niremberg is like our main figure here. So he was running a lab at the NIH, and he was going to work with, later he would work with like postdoc Heinrich Matthaei and Philip Leader. But he was taking in knowledge from all sorts of different places and all sorts of different scientists. And so they suspected, and as George Gamow had proposed, so I was like, I'm not going to try to give like a conclusive history or anything. I just want to give people the credit where credit's due, when I know where the credit's due. But so he said pros, and then they would other groups would confirm in various ways that that there were 3-letter words, but they didn't know which one spelled for which amino acid. And so basically they're cracking the code is figuring out which words spelled for what amino acids. And so to do this, they were going to do this in a couple phases. And the first was kind of like a brute force approach to cracking the code. And then the other was getting a little more nitty gritty. So at this time, where do things stand when he's starting his experiments? So basically, they knew some things, but not a lot of things. And so some of like the existence of the RNA intermediary between the DNA and the protein. So we're talking about the messenger RNA here. And so I'll show you, they actually did some really cool experiments that were investigating the components of this that were important. And they were doing this in a cell-free manner. So they would like break open bacteria and then isolate different fractions of it and then mix them together to see what you needed. And so there was evidence that there was some sort of messenger RNA before the works that we're going to talk about today. Some of it came from Alfred Hershey, who was the same like Hershey-Chase experiment guy, who showed that with Martha Chase that if they labeled nucleic acid of bacteriophages, so these viruses that infect bacteria, the labeled nucleic acids would get into the bacteria and the bacteria get infected, and it was this genetic information that was coming from the nucleic acid instead of the protein. But he also did some other experiments. For example, when he infected E. coli with the T2 phage, he found that there was some RNA that was actually made and degraded. And other scientists, Bulkin and Astrachan, found that the temporary RNA that was made was actually complementary to the phage's DNA and not E. coli's. So the phage was getting the E. coli to make more phage nucleic acids, and this nucleic acid was RNA, and it was temporary. So this was some early evidence of the existence of messenger RNA. So it's just some of the evidence. So basically, scientists strongly suspected that template RNA was being made and somehow read and understood by this protein making machinery, but they didn't have the translation dictionary. And so this is where these experiments come in. And so there's going to be several classic experiments. This is like one of the early super duper classic one. This PNAS article, the dependence of cell-free protein synthesis in E. coli upon naturally occurring or synthetic polyribonucleotides. And so this was done by Marshall Niremberg and Heinrich Matthaei at the NIH in 1961. And so basically in this important paper, they established a cell-free E. coli expression system where basically they're able to break open E. coli, separate various parts, so separate the ribosomes, separate what they call ribosomal RNA, which is confusing because that's actually like the template RNA, not the actual RNA making up the ribosomes. There was soluble RNA, they called soluble RNA, something called soluble RNA, which was tRNA, so transfer RNA. So that's confusing because they call that sRNA, especially when you work on small RNAs, which we abbreviate sRNAs. That got really confusing. And then also like the various other components of the cytoplasm that are needed, and ATP and various things. And because they're able to separate these components, then they're able to like add in various messenger RNAs from other species or from synthetic template RNAs. And so in their first phase of this genetic code cracking, what they do is they take their mixture. And so they take this mixture and it has the ribosomes. And so it has all of the other stuff. So it has the like soluble tRNA, the soluble RNA, which is the tRNA. And so it basically has everything except for the template RNA, which they refer to as like the ribosomal RNA, which remember is really confusing. But then what they could do is they could include stick in different RNA instructions and see what it coded for. So this would work they stuck in like a sequence that had like a single RNA letter. And in this way, it wouldn't tell them like, was this was the codon one letter? Was it two letters? Was it three letters? Was it four letters? But they didn't know. And so this way, no matter what length the codon was, you wouldn't have to worry about like overlapping things or what order it's read in. Basically, any codon that was a single letter, one of these letters would make an amino acid. If this was really how things were working. And so this was, it was a lot easier to start with these because it's like unambiguous. So what did they do? So what they wanted to do was they wanted to see whether protein was getting, what amino acid was getting made. So they included a mix of amino acids where one at a time was radio-labeled. And then they measure radioactivity and compare it to the amount of total protein made. So there's always going to be something made, but you're only going to see it if it's labeled. And so they have to test a mix of each with the amino acid labeled. And when that amino acid is labeled, it'll be high. The radioactivity will be high. And so I kind of like to think of it as, I don't know if I'm like a total Friends person, that episode where Monica's trying to figure out what the heck the light switch goes to, and she's like flipping on and off the switch and checking all of these different lines and stuff, but it turns out that it's actually turning on and off the TV in the other room. So Phoebe's watching TV and thinks that she's controlling the TV by blinking, and really what's happening is that the light switch is controlling the room in that one. So the point basically is that you can only be, you're flipping the light on in a room, but you're only going to see it if you're actually like in the room. So you're only going to see something labeled if you're only going to see a product made if you have it labeled. And so because you don't know what you're going to be labeling, you have to try this out for each of the different letters. And what they found was that if you put in poly-U, then polyphenylalanine will always be made. But it will only be radioactive and therefore detectable in the setup where it's labeled. And so if you label alanine, you're not going to see it. If you label arginine, you're not going to see it. If you label asparagine, you're not going to see it. But if you label phenylalanine, now you're going to see this increase in radioactivity. And so you can see in this paper, so if they put in poly A, and if the phenylalanine was labeled, then they got a hit. And so here you can see that they have radio-labeled amino acids with C14. And so here phenylalanine is labeled. And if they add polyuridylic acid, so if they add that poly-U sequence, now they're seeing a high amount of radioactivity. But if they labeled any of these other amino acids and they added poly-U, they weren't getting the high radioactivity, just like some background stuff. And so if they put in radio-labeled phenylalanine, they could only get it made if it's poly-U. They put in poly-U. So if they put in other sequences, they don't get the poly-U incorporated. So this is telling you that poly-U was coding for phenylalanine, and specifically for phenylalanine, and phenylalanine was specifically coded by poly-U, nothing else. So you might be wondering, like, how did they get these ribosomes to make peptides or make proteins when normally for that to happen, you need to have a start codon. So you need AUG, which spells methionine, or start if it's at the beginning of a chain. So it tells the ribosome where to start. But they were using like poly-U and that sort of thing. So how are they doing that? Well, it turns out that they got kind of lucky in that if you use a high concentration of magnesium, you can bypass the need for a start codon. So magnesium is able to kind of like help, so it's like positively charged, it's able to help kind of neutralize the negative charges of the phosphate backbones of the RNA molecules. So when you're bringing all this negative charge into close contact, because you have all this ribosomal RNA, you have this tRNA, you have this messenger RNA. It's a lot of negatively charged phosphate groups. And so the magnesium is able to kind of keep them from repelling one another. And then once they get close and they can find that codon, anti-codon match, then they're more stable, even though it's not an AUG. And so, yeah, so if you're confused about that, it's the magnesiums at work. So that was really great. So we figured out poly-U, and this is like super, super duper important, not just because it's like poly-U, but because it's like directly showing a link between the DNA sequence and the protein letter. So it's like, boom, it's like so important, but it's only a single letter. And so they were able to do similar things with the other polys. Like poly C and poly U. They didn't do poly G because it made these weird knots and that sort of thing. So they could figure out 3 letters, but what about all the rest? And so now we need to do some thinking. So what the problem is that if you have, especially if you don't know if it's like 2 or 3 letters, then you wouldn't know if the sequence is going, what the heck the sequence is. So if you have this like random sequence, you're not going to know what the sequence is. Even if you have like put in a mix of U's and G's or something, you're not going to know the sequence. Even if you have like an ordered thing of them, you don't know if it starts here, or it starts here, or it starts here, or it starts here. So we need to really control this down to the smallest piece we can get. And so they did some experiments. So this is actually from the later Nobel Prize lecture. And so here you can see that they and others found that you really need at least 3 letters. And so this is using a different method I'll tell you about in a minute. But so you can see that only when you have 3 letters were you able to get a tRNA to bind. And so I'm talking about tRNA binding because we're moving into this second phase of the experiments, the second type of experiments. And so now we're talking about the tRNAs, which they're referring to as soluble RNAs or sRNAs. And so the role of this tRNAs was still pretty inconclusive. So there was a lot of competition going on at this time, but there was also some nice collaborations. And so Lippmann and Nathans gave the Niremberg group purified transfer enzymes. So these tRNAs, and they showed that phenylalanine tRNA is required for phenylalanine synthesis, and that transfer enzymes and GTP were also needed. And having this tRNA connection gave them this other idea. So basically, some other scientists' experiments, Arlingas, Fabriklux, and Schweet and Kaji, and Kaji have found that poly-U causes tRNA loaded with phenylalanine. So this phenylalanine charged tRNA to attach to ribosomes before a peptide bond was actually formed. So before the amino acid got linked to the growing chain. And so this got them thinking. So you could get this tRNA to bind to the ribosome. And so if the tRNA is theoretically only binding to the ribosome, if the corresponding codon is bound to the ribosome. So could they add ribosomes, add tRNAs, so load ribosomes with like specific sequences, add radiolabeled tRNAs, and then mix them and see if they bind. And they're able to see if they bind using these filters that the ribosomes can't get through, but the unbound RNAs can. And so if they're bound, then if they're bound, they're going to bind to, and then you'll see the radioactivity on your filter. But if they are not complementary to one another, then they, so if they don't like match, then you're not going to get the radioactivity trapped. And so you can radio label different tRNAs, and you can use different sequences to try to tease apart these different combinations. And so this is what they did. So this is another one of their classic papers, RNA code words and protein synthesis, the effect of trinucleotides upon the binding of sRNA to ribosomes. And so this was Niremberg and Leader in 1964, and they're showing this technique, this like filter binding technique. And So you're going to see an increase in radioactivity on the filter when the tRNA loaded with the radioactive version of the corresponding amino acids present. So once again, we're labeling with like C14, but this time we're labeling the amino acid on the tRNA. So it's already like charged with it. And so basically, so this, if they don't label anything, so this is when it says like none, so this is the polynucleotide that they're adding. So this is like the template they're adding. If they don't add any, anything, then they don't see any activity. This is just like the baseline. If they add poly U and they have this is showing which one is labeled. So if they have labeled phenylalanine, they get a signal, but if they have labeled lysine or proline, they don't. With poly A, if they have lysine labeled, they get a signal, but if they have phenylalanine or proline, they don't. And with poly C, if they have proline labeled, they get a signal, but not as much with lysine or phenylalanine. This one's a bit less. But so it's that this one wasn't like quite as clear here. But so now they kind of needed to, this is still using like single letter sequences, but really what they need to do is now they need to be able to use the like defined sequences. So now we get back to that problem of being able to use just the codon, which means that they need to get all of those possible 3-letter combinations. And at this time, they couldn't just like go order them like we would today. So they had to do some harder work. And so they would get some help from like Leon Heppel and Maxine Singer, who would advise them. But they took advantage of some enzymes that were discovered even by like some of their competitors, like Severo Ochoa and his big lab at NYU. And so they had discovered this polynucleotide phosphorylase that was important for some of the stuff that they were going to do. And so basically what they needed to do was they needed to get those 3-letter sequences. So it turned out that they could get some of them if they had like a poly UG sequence or a poly something sequence where it was like just a combination of like 2 letters, then they could kind of like cut those up and then separate the pieces and separate the pieces by their like chemical properties and then test those pieces. But for other of them, basically what they did is they ordered all of the ribonucleotides, so each of the like 16 2-letter nucleotide combos from this German company that was advertising them in a journal. And then they used this polynucleotide phosphorylase to add a 3rd letter onto the end of those. And so this was the phosphorylase that they got from the Ochoa lab. And so by this way, they could make all of the different 3-letter combinations. And so they were able to get the code mostly cracked by 1966. So here is one of the papers from 1965 where they have, I'll show you more in a minute, but they've done this for most of them. And then in 1968, they were awarded the Nobel Prize in Physiology and Medicine. Niremberg was Niremberg, Hawley, and Corona, who also made key contributions. And so I just want to show you a little more of those papers because they're really cool and elegant. Okay, so this is the first one. And so you might remember, if you've been following, I did a talk, a post on like cell-free protein synthesis a few days ago. So check that out if you want to know more. But a lot of the groundwork was really laid by this work. So basically what they did was they took this, they took E. coli and they separated the various fractions and then were testing them. And so they basically, it's confusing because we're talking about like the ribosomal RNA, which is the messenger RNA, the template RNA. So basically they took separated the ribosomes from like the cytosolic stuff, so the like general cellular stuff, so they could like sediment out, like precipitate, or like based on the weight, these ribosomes are like huge and bulky and stuff, and then they could separate out the ribosomes from the like template RNA, and so they call this like the ribosomal RNA, which remember is really confusing. Then they have the cytosol fraction, and in the cytosol fraction, they have like soluble RNA, which is like the transfer RNA, the tRNA, but they're calling it soluble RNA or sRNA. So they take this labeled amino acid, in this case valine. They don't really care what it is. They just need one, because theoretically, if you're making a protein, the protein's going to have an amino acid, any amino acid, and so they just have to label one of them. And so they labeled valine. Not sure why. Maybe it was cheapest. But now they have, or maybe it was more frequent, I don't know. Anyway, so if they mix it with the soluble RNA, so with the tRNA, they don't get really protein made. But if they mix it with the ribosomes, now you do get protein made. And so now they just show it various amounts of ribosome. So basically they just want to figure out how to like what's needed for translation. And so they test these various things. They also they tested some like antibiotics and found that those were inhibiting the ribosome, which a lot of antibiotics do inhibit bacterial ribosomes. Then they tested various things. They tested, you need amino acids, that sort of thing. So that you can see too that they tested like various amino acids, and so they see that they're all getting incorporated, which is consistent with proteins getting made. And they also, what's really cool here is now they're testing, okay, so we know that you needed that template. We know that you needed the ribosomal RNA. So they did a bunch of like experiments showing that you needed the ribosomal RNA, and it couldn't just be DNA, or it couldn't just be whatever you want to stick in there. You needed that ribosomal RNA, which is the messenger RNA. And they showed too that you could replace it with messenger RNAs from other species. So they put in like yeast RNA, some virus RNA, and various things, and they got protein made, which is pretty cool because now you're seeing, okay, this genetic code is universal. No matter what, you can stick an mRNA from other species and you still get protein made. And what's really, really cool, now this is the part that we were talking about with this beginning of cracking the genetic code, is that when they stuck in poly-U, now they got phenylalanine incorporation. And so this was really awesome because this is showing that you need this, that only poly-U would make phenylalanine. And that only phenylalanine would get made if you have poly-U. Then they just show, it really is this thing and stuff. So basically, this was like the start of it all. And so this is the paper where they're showing the filter binding experiments. And so here, basically, they come up with this way where they can figure out which amino acids are coded for by various codons, or at this point, it was still just like a poly sequence, and they were testing like various effects of like heat and that sort of thing. So this was another important one. Then they were further expanded upon all of this. And so here we're seeing them describe some of all of these different experiments that they did, testing all of these different trinucleotide combinations. You can see that they use different synthetic methods. So some of them were like a ribonucleotide digest of the poly-UG. Some of them were the isolation, some of them were like these polynucleotide phosphorylase with the two sequences and various things. And so, they did a lot of work. And so you can see that they tested all these different combinations where they have different amino acids labeled, and then they have different trinucleotides added, and then they're looking to see what's getting caught on their filter. And so you can see that in cases where something gets caught, you're going to see a bigger number. A lot of these are negative because I think it's just because they're subtracting out the background. But you can see that when you have, like, they put in bold the ones that are actually like coded for by that thing. And then they summarize all of this data in this like codon table, which is pretty awesome. And they talked a little bit about the significance and stuff. Later on, they would also, so there was some like weird stuff going on with a couple of them where they didn't really seem to be coding for anything that they could detect. And so they were suspicious that these might be like termination codons. Later, they would do more experiments actually showing the termination codons and the like what's required for this termination. And so this other group had found that you need this like R factor for the release. And they were able to do these experiments where basically they bind the tRNA with like bound to a modified methyl group, and then they get it to bind the ribosome using the start codon AUG, and then they add the stop codon and get this release factor and get the measure the release. So what's still left on and what's released from the group. And so yes, that was more on like tying up the ends. You can learn a lot more about it. His Niremberg's Nobel Prize lecture is really good. And oh yeah, so they added like DNAs to the mixtures to like both reduce viscosity and also so that you wouldn't get messenger RNAs getting made that you didn't want. You can learn a lot more about like the history of this all too. This nice commentary, the RNA code, Nature's Rosetta Stone, which was done by Leader and Kasky in 2014. And so those are just some of the sources that I was getting some of the more historical aspects of things, and less elegant, but I also have a post on my blog. So I'll link to all of this sort of thing. And thank you for letting me geek out.